are not isotopic in nature. These are magnitudes of fluxes here. So net flux is different than the isotopic signature of the net flux. The disequilibrium is the difference between the one-way fluxes in the signature. That disequilibrium is a necessary piece of the puzzle along the way to coming up with these fluxes. But what this shows is the land sink and the ocean sink have increased. That's a real bonus for us. That is mitigating the CO2 in the atmosphere, and it's mitigating temperature rise. And we are uncertain if this will continue well into the future. And in fact, when you try to predict what CO2 is going to be, CO2 magnitude in the future, the uncertainty of the terrestrial biosphere models is among the largest uncertainties in trying to predict future CO2. Okay? The biggest uncertainty is what are people going to do? The IPCC deals with this by saying, okay, if we do business as usual, or we cut emissions by this, or we really cut emissions, these are the scenarios we work with, and then we move forward and try to investigate the other aspects of the system. So this, on the right, this is the same thing. It's just 1840 out to now. If you don't smooth out the decadal patterns, and you look at this is a really nice figure that comes from this, I think it's the Raupak paper here, but it shows up in the IPCC. So since 1750, we've got a certain understanding of emissions. And those emissions, which are repeated here, these are divided into two things. There's the fossil fuel bit, and then there's deforestation. That is balanced by the sinks, okay? And the sinks include the ocean, which is dark blue, the land biosphere, which is green here, and then the atmospheric increase is in between. Okay, there are three places for it to go: the oceans, land, or stay in the atmosphere. And you note that the variability in the biospheric sink is really what leads to variability in the atmosphere. It's not variability in the ocean sink. The ocean sink is primarily an ocean disequilibrium, a carbonic acid reaction with a weird atmosphere. And so that takes time for it to happen, and it's happening, and it's continuing, and it's a fairly consistent kind of thing. Okay, questions about this? This is the final product of the application of mass conservation to C13 in the atmosphere. This is the kind of thing that we can learn by looking at that and studying the processes. So I'm going to skip the next few so that we can cover some other stuff. You can read through it once you get the notes. I want to highlight without any detail that we published a paper a couple years ago that was focused on this disequilibrium. And that pattern that I showed you of respiratory fluxes being more enriched than photosynthetic fluxes doesn't appear to show up at the forest where we really studied this in detail. It's actually the opposite isotopic sign. And there are some reasons for this, and we're still trying to sort it out. But if you're interested, you can check out this paper, or I can talk to you about it. So with the time that we have left, I've got two more topics that I want to cover. Again, I apologize for the notes. I mean, it's a bummer that you don't have them. But let me talk more. All of this stuff has been global carbon cycle, which is what goes into the introduction to my grant proposals. But I don't really work on any of that stuff. I work on this stuff, which is, I work on flux towers, so towers that are built in forests or in grasslands, and we directly measure the exchange of CO2 or of water vapor between these ecosystems and the atmosphere. And then we measure isotopic variation on all these things so that we can try to understand the processes. So I really am interested in understanding small-scale forest carbon cycling as opposed to the whole globe. And when I say small scale, my department is a biology department, and they think I'm all out there on the big end because they work on cell biology, right? So a lot of factors influence CO2 that you might measure within a forest. Okay, there's photosynthesis and there's respiration. Respiration occurs in the mitochondria of plant tissues. It occurs when animals eat plant tissues. It occurs when decomposers eat organic material in the soil, but it all begins with photosynthesis, right? This is the energy for all these other processes. And this is just the normal activities of all of these living organisms eating dead things or eating live things. So there's CO2 uptake by photosynthesis, and then there's release by all of these other organisms that are there. And if you look at patterns of CO2 in the atmosphere in a forest, this is what you see. This comes from a study in the early 90s Graphics have changed, as you can see here from this photo. That's a tree. That little diamond is supposed to represent a tree. On top we have time, and so this is midnight, and this is midnight, and this is the middle of the day. And this is CO2 on this axis. Note how low it is. Okay, middle of the day is about 360, so this is the early 90s. It's gone up tremendously since then. H here corresponds to one canopy height, which is about 12 meters. And so 2H is twice the height of the canopy, and H over 2, et cetera. Those fractions are within the vegetation canopy. And what you can see is during the day, there's very little difference in the CO2 measured with height. At night, there's a big difference, beginning at dark and building up overnight. This is a profile, a height profile of the same. So it's height here, and this is CO2, and time is represented by the different lines there. And so this is night with the highest buildup in CO2 near the ground, and this is day, still with a little bit of buildup near the ground, okay? But big differences between these. Okay, so photosynthesis is occurring during the day only. Respiration is occurring all the time. The patterns that you see here are not driven by just photosynthesis and respiration, though. The big buildup that you see at night is caused by respiration, but also by changes in the stability of the atmosphere. And then the decrease that you see from night to day is not caused by photosynthesis. It's actually caused by a change in the atmosphere. This is a fair weather conceptual diagram of how the atmosphere mixes. And let's begin here. So this is height in the atmosphere. Let's begin at sunset. The sun has been heating the land surface, and that generates buoyancy, and it leads to mixing. And when that stops, when the solar heating of the land surface stops, there is a radiative exchange. Imagine that now that it's dark. There is infrared exchange. The sky is cold, the land is warm, there's a net loss of energy, and the land surface gets cold through radiative loss. And if you ever sleep in a sleeping bag outside and you got your arm sticking out and your arm gets really, really cold, it's for this reason, right? This is a radiative exchange. So this means that air that interacts with the land surface becomes cold, and cold air is dense. And cold air, because it's heavier than warm air, settles to the ground. And this leads to stability in the atmosphere. This is referred to as a temperature inversion. And so at night, there's a very stable nocturnal boundary layer that is tens to hundreds of meters deep. And respiration is dumping CO2 into this shallow atmosphere, tens to hundreds of meters deep. And that may actually grow over the course of the night. And then the sun hits it in the morning and begins to generate turbulence. And as it generates turbulence, the depth of what's referred to as the convective boundary layer really grows over a couple of hours, very, very quickly. And it mixes in the air that was above that stable nocturnal boundary layer. And that's yesterday's mixed layer, which is low in CO2. And so this mixing here pulls in a bunch of low CO2 air into the forest and decreases the CO2 that's there simply because of mixing. Photosynthesis is occurring, but it's a tiny flux compared to this, what's referred to as an entrainment flux in the atmosphere. And then during the day, when the sun is really heating the land surface, this leads to really vigorous mixing, and you get big eddies that traverse a couple of thousand meters into the atmosphere. So you all experience this when you fly on an airplane. You take off, and it's really bumpy for the first 10 minutes of a ride, and then eventually you can take off your seatbelt. That's because you've left this bumpy, turbulent, convective mixed layer, and you are up out into the free atmosphere up above it. And when you fly out of here, and most of you are probably going to fly away, we got a bunch of fires in Utah right now, and you're going to be able to see this layer because the smoke is making its way primarily only into this convective mixed layer. So you ought to be able to visibly see the change as you move through this layer when you fly out of here. So the same pattern that I showed you can be observed on a tall tower. This is a 500-meter tall tower in Wisconsin, and these are averages for July of '97. And what you can see here is at night, build up at different heights, and that buildup reaches a max, and then when the sun comes up in the morning, all that stuff from up above gets mixed in and it comes down. And things are fairly well mixed across this big vertical gradient, okay? But then there's a slow decrease in the CO2 over the course of a day. That slow decrease is the result of photosynthesis. Air is moving across Wisconsin, the forests are slurping CO2 out of the air, and it's decreasing CO2 over time. So this phenomenon happens not because of the presence of a vegetation canopy, but because of the stability of the atmosphere. These measurements are well above that 30-meter tall vegetation canopy. Okay, so we're supposed to be thinking about isotopes. I took you through that pattern so that you can then understand what the isotopic patterns are like. This green box is supposed to represent a leaf with one stoma here, or represent all the stomata. So you learned from Jim, CO2 diffuses through the stomata. There's a diffusive fractionation. There's an enzymatic fractionation. The net result is that atmospheric CO2, which has some delta value, ultimately turns into leaf biomass that is more negative, or sugars that are more negative, et cetera. Photosynthetic discrimination. When this happens, there is a removal of the light isotopes, more negative, and that leaves the air that's left behind more enriched. You take away the isotopes, the light isotopes, and what's left behind has relatively more of the heavy isotopes. When that organic material gets respired by the plants themselves, or organic material from the plants that gets eaten by something else, there is very little fractionation, and so this takes this depleted C13 and returns it to the atmosphere. So the net process of respiration, when CO2 is building up in that forest at night that I've just shown you, it's building up with a depleted signature because respiration is adding this depleted stuff. A long time ago, this said no fractionation during respiration. People have sorted out a lot of important details that are associated with respiration. Now, that no fractionation in respiration actually came from a postdoc that was working with Jim here, paper in 1997. They took isolated plant protoplasts, so these are plant cells that don't have the cell wall, and they put them in test tubes, and they fed them C3 or C4 sugars, and they looked at the respiration that came from them. And they showed quite clearly that there is no fractionation associated with mitochondrial respiration. So if you think about the Rubisco fractionation, this is an enzymatic thing that's associated with a biochemical process. And mitochondrial respiration is a complicated, the Krebs cycle, right? There's a whole lot of enzymes involved. Those enzymes don't have the same kind of fractionation that Rubisco does. And that's a fundamental nature of the process. Those enzymes just don't fractionate. So if all of the substrate gets respired by a cell in a test tube, then there is no fractionation. But that's not the way that respiration works in a living organism. Take a 6-carbon sugar, chop it in half of it goes into respiration, and the other half turns into some kind of carbon skeleton to build up other molecules. And so because there are molecular positional differences in the isotope ratio in sugars or in other molecules, there are lots of fractionations that are associated with respiration because of the complicated link between respiration and biosynthesis. So Kate's going to show you on Friday about biomarkers, and the reason that there are signatures in biomarkers is because of all of these very important fractionations that occur within an organism. This is an example of metabolites within a leaf, and they are being compared here to the bulk leaf, the dry bulk leaf carbon isotope ratio. And so that's 0. And you can see here that these different compounds are a little more enriched than the bulk leaf material, and these compounds are more depleted than the bulk leaf material on average. These are all within a plant, within a leaf. If you look within an ecosystem at different pools, in the same kind of fashion comparing to bulk leaf material, these different pools here have consistent systematic isotope ratio differences because of these fractionations. Okay, so if you're interested in studying these processes related to the trophic ecology of fungi or carbon allocation between plants and other organisms, these labels can be very, very useful. Ultimately, they're quite complicated. This is from, you probably didn't read all of the readings because it was a really huge one, but if you did, this is one of the readings that I had suggested. This is a kind of a conceptual diagram of all the things that might lead to fractionation within the plant soil system. And there's a lot of them. And so this came from a conference where somebody's up on a whiteboard and they're kind of spinning their wheels and thinking about it. We understand some of these really well, and we have really almost no idea about how others work. But it's a complicated kind of system. And again, if you're interested in studying the carbon cycle, carbon allocation within plants, carbon cycling between plants and other parts of an ecosystem, these are all quite important. But those fractionations can't be sustained over long, long time periods because of conservation of mass. You got a certain amount of carbon coming in, there's carbon that leaves. Ultimately, that has to balance, or maybe it doesn't quite balance. Ultimately, the forcing on the atmosphere is you don't have to pay attention to any of these processes. And so the net result of respiration is that respiration adds CO2 to the atmosphere and depletes the C13 in the atmosphere. Photosynthesis removes CO2 from the atmosphere and enriches the atmosphere that's left behind. And so this is the signal that we find if we go looking for it in a forest.